This is a quick video that will hopefully help you understand the resting membrane potential, what contributes to it, and why it is necessary for cells to function. So here are some important contributing factors. We have sodium-potassium ATP pumps. So these are pumps requiring energy. ATP must be used. These pumps will pump 3 sodium outside the cell and at the same time pump 2 potassium ions in. You also have potassium leak channels, sodium leak channels. Both of these channels are open at all times, and potassium will slowly leak outside the cell and sodium will slowly leak inside the cell. You also have negatively charged proteins. Usually these are along the inner membrane of the cell, but they may be within the cytoplasm. The proteins cannot exit the cell, they're too large. All of these will play a part in establishing a resting membrane potential, which is between -50 and -100 millivolts for cells. For nerve cells, neurons, it's -70 millivolts. Also, because of the pumps that are present here, there will be concentration gradients established for sodium and potassium ions. What I've drawn here is a simple diagram to illustrate the different factors that go into the resting membrane potential. We can call the outside of the cell the interstitial space, or we can say extracellular. The inside of the cell we can refer to as intracellular, or we can say cytosol, which is the fluid portion. And then I've also drawn out here just a simple plasma membrane, and I've included in that plasma membrane some leak channels. So here's the first one. Here's a sodium leak channel, and what will happen is this is always open and sodium continuously will leak into the cell. It does this because there's a higher concentration of sodium outside the cell than what is inside the cell. The next leak channel here is potassium, and potassium will continuously leak out of the cell, and it does this because there's a higher concentration of potassium inside the cell than outside the cell. Remember, leak channels are open at all times, so you continuously have sodium coming in and potassium going out. You also have a sodium-potassium pump, and that's right here. This is an ATP requiring pump, so it takes energy to operate, and what it does, it will continuously pump sodium out, 3 sodium, and potassium in, 2 potassium. So the pump is what generates these concentration gradients. Sodium gets pumped out, it continuously leaks in. Potassium is pumped into the cell and it continuously leaks out. But at all times you maintain these concentration gradients with more sodium outside the cell than inside and more potassium inside the cell than outside the cell. These concentration gradients are a form of potential energy. I've also put in here some negatively charged proteins. Here, here, here. A lot of the proteins in the cell are along the inside of the cell membrane, but some are also in other places of the cell, but they are negatively charged and they do not leave the cell. So if you add up all the different charges, electrical charges of the sodium, potassium, the negatively charged proteins, what you wind up with is a potential difference between the inside of the cell and the outside of the cell, with the inside of the cell being negative compared to the outside. And we call that the resting membrane potential. For neurons, it's about -70 millivolts. The other thing I've drawn here is a ligand-gated sodium channel. So this is present on some cells, including neurons. I'm using that as the example here. A ligand is anything that will bind to a receptor. So this little diamond here I've drawn is the ligand. When the ligand binds to this gate, this is a gated channel, it is usually closed. When the ligand binds, the gate opens. So the gate is now open, and because you have a lot of sodium outside the cell, sodium rushes in. So now you have more and more sodium coming into the cell. Sodium has a positive charge to it. Because of this positive charge, the resting membrane potential is going to go toward zero or in a positive direction. We call that a depolarization. And it will eventually reach a positive 30. This is how an action potential is initiated. I haven't drawn the rest of the action potential because that would get into more gates, and we're not there quite in the chapter. The reason that sodium rushes into the cell is because there is a concentration gradient. This was potential energy. Once the sodium starts moving into the cell, it is now kinetic energy, and that energy is actually going to be electrical energy. Within cells, it's the movement of ions that translates into an electrical current. So just to summarize a few things, ions move down a concentration gradient, releasing energy. The ions that we've talked about, sodium and potassium, have that concentration gradient because of the sodium-potassium pump that required energy. When you form a concentration gradient, you're storing energy as potential energy. In neurons, when gated channels, and that was the ligand-gated sodium channel I demonstrated, the ligand binds and the sodium quickly moves into the cell. That's what changes it to a positive potential. That's the initiation of an action potential. You've now converted the potential energy that was stored in the concentration gradient to electrical energy. That action potential will continue to form with other gates opening, and we'll go into that at a later time. If you don't have a negative resting membrane potential with those concentration gradients, neurons and muscles will not work. So I hope this has helped you understand a little bit the resting membrane potential, how it's formed, and why it's important. In later chapters, we'll go into the rest of the formation of the action potential and how it's propagated down the axon.